All right. Good morning, everybody. Thank you for coming. It's a pleasure to have today with us Dr. Spencer Smith from University of North Carolina. Dr. Smith did his undergraduate at UCLA and got his degree in physics and mathematics. He studied neuroscience and neuroengineering at UCLA, where he got his PhD as well. And he joined the UNC-Chapel Hill Neuroscience Center. Neuroscience Center in 2011. And in fact, the reason I was a bit late, he was describing to me one of the devices that he built. He does a two-photon electro-optics to actually visualize brain activity. And so what he's going to be talking about today is one aspect of that system. Thank you for coming.
Okay, thank you. Thank you very much for that introduction. It's my pleasure to be here. This is actually the first time I've spoken to an engineering audience, so this will be fun. Hopefully different questions. You can feel free to speak up and interrupt me at any time. I'm happy to go into any depth or answer any questions along the course of this. So my lab is fundamentally a neurobiology lab. We study how individual neurons and the networks they form compute. And in particular, we work on the problem of how different parts of the brain work together. So we study this at multiple levels. We study this at the level of multiple area circuitry, naturally, because that's where our problem is defined. We're also interested in local circuitry because we want to know how inputs from other cortical areas will modulate the function and signal processing in other cortical areas. And we're also interested in cellular mechanisms. And I won't talk about this much today, but this is the reason we're interested in cellular mechanisms, that we're interested in how activity in different cortical areas is coordinated during behavior, and cellular mechanisms are a big component of that, and understanding how those are engaged is important to get a clear picture. And in the course of this, and this is what we're going to talk about today, we have to overcome technological limitations so that we can do better experiments. So we, part of the lab and part of what we spend our time working on are generating new instrumentation to explore untapped technological headroom to perform better experiments. So what circuitry do we examine? We examine visual cortical circuitry, and that's where all of our studies are founded. And I'm interested in visual processing because it's a very computationally interesting problem. Moving from pixel data, as one might get from photoreceptors, to a perception of the world is tremendously complex. And our best computers and machine vision algorithms still struggle to do this in even very controlled circumstances. But evolution has completely nailed this problem. Even insects can navigate via virtual landmarks better than self-driving cars can. So one of the things that we do, since we use mice as our model system, is we try and understand what mice can do with their visual system. So mouse eyes are very, very small. The spatial resolution of their vision is very, very poor. However, they can do a lot of the things that we take for granted as well. So they are experts at navigating via visual landmarks. And one of the things that we wanted to quantify in this particular example is their global motion perception. So this mouse is nose poking to the side of the touch screen where the dots are moving up. And without cueing the mouse, we're going to switch to a testing block where we decrease the coherence. So a large fraction of the dots are moving in a random direction, and the mouse still has to nose poke to the side where the dots are moving up. Got that one right. That was 20%. That was very hard. There was probably some luck involved with that. 70% is a little bit easier. Gets that one right as well. When he gets the right answer, he gets a little reward in the back of the cage, just a sweet milkshake thing. So they'll do hundreds of trials like this. And by varying the coherence, we can figure out where their performance drops off. So we can test the limits of their perception. This is almost exactly the same kind of test that they can give to non-human primates and humans. And this is something that this selectivity to this type of stimuli is something that occurs developmentally. So people who are born with cataracts in both eyes, which is actually not that uncommon, but in the developed world, the cataracts will be corrected very early after birth. In very rural parts of the world, people may get to be 12, 13 years old before they actually have their vision restored. And for those people, they do very, very poorly on this test. Their coherence thresholds would be shifted 30 to 40% to the right. So they need a much larger fraction of the dots all moving in the same direction for them to be able to perceive the coherent direction of motion. So this is one thing that we're doing. We also are looking at naturalistic images in mice. We're trying to get a feel for how, what the limits of their perception, what their visual system can actually do. And then to actually study what the neurons are doing in their brain, we use two-photon imaging. So it turns out that calcium indicators fluorescent calcium indicators provide a very high fidelity readout for neuronal spiking. So this is a movie, two-photon movie. Each one of these little dots that are lighting up are individual neurons. This is in a living mouse viewing visual stimuli. In this case, we're looking at an organic dye called fluo4. So this is a dye that I've loaded into these cells. And this scale, so this is about 15, each one of these cell bodies is about 15 microns across. So this is a pretty small field of view. This is maybe, a few hundred microns, well, probably closer to 250 microns across. Very small field of view. These are blood vessels, the black regions that you can see here. These are some glia, some support cells. And we're using two-photon imaging. Two-photon imaging is a nonlinear imaging modality. Probably lots of you are familiar with it. What's special about it is that you only get excitation right at the focal point. The efficiency of excitation falls off by the square of the intensity, and so with normal one-photon fluorescence, it falls off by 1/r, and so you get this kind of cone of dim fluorescence above and beyond the focal plane. And in two-photon imaging, you only get excitation right where you're stimulating. Yeah.
What we observe here is a stationary mouse, right? It is not a moving mouse.
That's right. So this mouse is head fixed. So this mouse has had surgery to open up a window into its brain. So we take off a little bit of scalp, a little bit of skull, put in a glass cover slip, super glue that in there. There's also a metal, there's different ways to do this, but we also have a metal implant, and then the mouse is secured underneath the microscope for the duration of the experiment. This particular case was a lightly anesthetized mouse. We've also done this in awake mice. You just have to habituate them to be sitting there head fixed. They're not in pain at all. They're after this surgery, they recover extremely quickly. Okay, did I answer that question? Okay. So what's special about two-photon imaging, the reason we use this particular modality is that we need to focus in hundreds of microns into the cortex. And if we had out-of-focus fluorescence, we'd be getting very low signal-to-noise because most of our fluorescence photons would be coming from dye molecules that were up above our plane of focus. So by using two-photon imaging, we can see deep into scattering tissue. If the brain was completely transparent, this wouldn't have been so much of an issue, but since most of the light that we're detecting, or maybe not most, but a large fraction of the light that we're detecting is scattered on its way back out, using two-photon imaging gives us a way to get high-quality, high-fidelity images, hundreds of microns into the brain. The caveat to this is that we get this very, very small field of view. And the reason that we want to see other fields of view is that so this paper came out in 2007. And what this is showing is the mouse brain. This is now a fixed piece of cortex. This was prepared by Andreas Burkhalter and a scientist in his lab, Quixin Wang, who's now at the Allen Institute in Seattle. They did triple dye labeling, basically just injected dye into 3 different spots in primary visual cortex and looked to see where the axons, where the outputs of these cells went in other cortical areas. So these V1 stands for primary visual cortex, PM, RL, AL. Those are just anatomical names. This is posterior medial, rostrolateral, anterior lateral. They're just anatomical descriptive names because we don't know what these different brain areas do. What was new in this paper is we didn't realize how well organized all of these brain areas were. We knew that there were projections from V1 to these other higher visual areas in the mouse, but we had no idea that it was this exquisitely organized. So when I saw this paper, I got very excited. I thought, man, wouldn't it be great to actually image activity in a patch of V1 and a retinotopically matched area in AL? Yeah, go ahead.
So did the colors show current currents and spikes? Is that essentially how they got this? So the spike occurred in yellow?
No, this is pure anatomy. So they injected dye, and that labeled cell bodies, and then that dye was transported down the axon. And just, yeah, so there's no activity marking here. In order to know where they did to do the injections, they did a bunch of single-cell recording, kind of classical, old-fashioned electrophysiology to figure out where to do the dye injections. But the data that you see here is purely anatomy. Connectivity, exactly, yeah. Circuitry. So, but unfortunately, I can't see both these things, right? So this is to give you an idea of the scale. So I'm going to shrink this down and show you about how big this is in V1. It's completely hopeless to image two different cortical areas at the same time. If we put on a lower magnification objective onto our microscope, this is a standard microscope, then we can get a larger field of view, but we lose our cellular resolution. If we don't want to lose our cellular resolution, then we need to go to much larger optics. So here's just another way to display that same thing. So this is an overview of the mouse brain and the different cortical areas. So this is primary visual cortex, higher visual areas, auditory cortex, somatosensation, motor output. And this is basically the field of view that we get with a standard conventional two-photon microscope. So if you go to Olympus or Nikon and give them $1 million, they can give you a microscope that can image a small little area like this. What we want to do is expand the field of view so that we can see these other cortical areas at the same time, so that we can image activity in both areas at the same time. You know, an alternative is you could put just metal electrodes into these different areas. The mouse brain is pretty small. You get more than 1 or 2 electrode arrays in there, and you're not really, you're disrupting a lot of the tissue. It's very invasive. With two-photon imaging, it's much less invasive, and you can image more neurons, and you can do it you can follow the same neurons over several days while the animal learns to do a visual task. We also want to be able to image activity in these multiple cortical areas at the same time because we want to know what kind of information is flowing between these different cortical areas. So what are both sides of the conversation that's occurring during visual encoding and behavior? And you know, the alternative would be to have that small field of view and then just move it back and forth. But since all the dynamics are happening on the time scale of tens of milliseconds, we don't really have time to move the preparation. I mean, the inertia involved or the masses involved are around 1 kilogram or more, and so we don't have a good way to move things that fast and have a settling time that would still give us the time resolution that we need, because we need it to relax within about 1 millisecond. So what we'd really like to do is just have a large field of view and then image in multiple areas at the same time. Unfortunately, to get a large field of view with high resolution requires large optics. This is just graphing kind of this problem out. Here on the x-axis, this is the back aperture diameter of the objectives to give you an idea of the size of lens that you would need. The field of view diameter, which is a function of the focal length of the lens and the scan angle, and I'm assuming a scan angle of plus or minus 5 degrees, the back aperture of the objective. And actually, this is fairly ambitious. In most commercial systems, it's closer to 1 or 2 degrees. But here we're talking about plus or minus 5 degrees. And then there's a family of curves here. Each one is, well, not curves or lines, but for different numerical apertures or resolution. 0.3, we can barely see individual cells. Right around 0.4 is where we can see individual cells. And you can see that the relationship is steeper for more moderate NAs. So if you want really high resolution, you have to go to much, much larger lenses to get even a small increase in the field of view. So we had to make a bunch of engineering compromises and find a sweet spot where we could make lenses that were large enough to give us a good field of view, but still be able to image individual cells. So we're going to be talking about kind of in this area of these families of curves. Okay. So in order to design these systems, fortunately, the software for optical design has rapidly developed, and there's really nice software for this. So this is a screenshot from ZMAX, which Mike Kudinoff taught us how to use, and glad Mike could show up. And what's nice is that you can constrain the search space. You can set you can define your system to remain within a number of parameters, And one of the parameters that you can constrain is you can have the search space only look for lenses that you can get stock, that you can actually order in and have delivered in a short period of time, rather than having the system search completely custom lenses. You can map out what your expected wavefront error would be. So what we need to do is keep you get aberrations as you start scanning large beams at high angles because lenses are not the same thickness at their center and the edge. By using these multi-lens systems, you can decrease your aberrations. And so this is just a plot of the expected aberrations. This is a plot coming out of Zmax for polychromatic light. We use ultrafast lasers for two-photon imaging. We use 9-10 nanometers, and the pulse width is about 150 femtoseconds, and so the bandwidth is about 10 nanometers. So we try and correct. So we're really only achromatic over this really, really tight region. But that's fine because all we care about is our excitation light. We don't actually have to generate an image with the emitted light, because in two-photon microscopy, we're just scanning a point along, and all of the green photons, all of the fluorescent photons that we get back out of it, we send to one detector. So we can have all kinds of aberrations in green light. We don't care. We just want to make sure that our excitation light can get down to a nice, tight focus. So this is a plot just showing that we actually have, we're expecting anyway for this particular optical system to be diffraction limited out to a scan angle of 16 degrees or a half width of 1.94 millimeters for half of the field of view. This is different than the optimizations that a commercial microscope manufacturer would do. Usually with commercial biological microscopes, you want really nice field flatness because you're looking at imaging specimens that are maybe just a small number of microns thick, and so you want really nice field flatness, and you want the system to be achromatic over a large range of visible wavelengths because you want to be able to do co-localization with maybe green and red fluorophores. We don't care about that. We don't need it to be achromatic in the visual. We don't need it to be, we don't need field flatness because we're imaging in living tissue that has all kinds of convolutions. If there's a little bit of curvature, we don't care because we're still hitting brain. The tissue that we're imaging is hundreds of microns thick, so we can deal with curvature. So we have some field curvature in our system. It turns out to be not that bad, even though we didn't optimize for this. Our F theta distortion is also not that bad, even though we didn't optimize for it. It just kind of came out of it as we were correcting for aberrations. It turns out to be not particularly significant, at least for our preparation. We measure a point spread function. So to try and make something brain-like, we embedded things in agar, which has nowhere near as many lipids and bilayers as what you'd get in the brain, but it's better than just measuring in water. So basically, we embedded sub-resolution beads. So these are, I think they were like 1 micron beads in fluorescent beads in an agar block, and then imaged through a cover slip. So we're trying to reproduce our imaging setup. And we imaged at 3 different depths, at 55 microns, 275 microns, and 550 microns. And by the way, the design that we were doing, we did everything assuming that we'd be focusing about 275 microns into the tissue, with the expectation that if we correct for aberrations at that depth, we're more likely to have good performance at more shallower depths as well. So these are our point spread function measurements. We're around a micron radially and around 12 to 13 microns axially. So that's well matched to the cell bodies that we're going to be measuring because they're around 15 microns. So we're small in X and Y, fairly large in Z, and this turns out to still let us preserve single cell resolution. This particular iteration of the scope is mostly stock lenses. We ended up, I'll show you the custom objective here in a second. This is a commercial objective that's on this right now, but this is some of the table optics, which I'll explain in a second. But this is mainly off-the-shelf lenses. This is just another view of the system. This is the custom objective, so this has a few custom lenses in it. So this gigantic achromat here in the middle, or doublet, is custom. The terminal element's custom as well, and there's one more custom lens in here. So then we have some off-the-shelf lenses as well, and this let us get images like this. So this is some data from that objective. You can see that we can resolve individual neurons. We can see dendrites. We can actually see even dendritic spines. So this is a 5-micron scale bar. These are submicron structures that we can resolve in these mice. We can image fairly deep. So this is the Z depth here. We can image down hundreds of microns. So it's 300, 400, 500 microns, 6, 7. And the cell bodies of these things are around like 750, 800 microns. So we can image deep. We do have good, tight point spread functions, even deep in the tissue. This is in the visual cortex. Yeah, it's all in mouse visual cortex. And then to give you an idea of the field of view, so I've shown you that we've got the resolution, and we can image activity. This gives you an idea of the field of view. So this is kind of calibrated left and right. This is the same system. We're zooming in on a data set that's undersampled. That's why it looks pixelated when we zoom in. But this is, not only are we getting two, I was hoping to get 2 or 3 cortical areas in the same field of view. This worked better than I expected. This is a 3.5 millimeter field of view, and that's enough to encompass V1 and all these higher visual areas, and even portions of somatosensory cortex all in one field of view.
What's the stimulus here?
The stimulus here, the animal's looking at a naturalistic movie.
Or what?
A naturalistic movie. So we've got one of these first-person videos. It's actually of a mountain biker going down a trail, which is not naturalistic for a mouse, but it has a lot of optic flow to it, and that's why we wanted to use that stimulus. And that does a pretty good job of driving activity. In this data set, we get a ton of cells. So these are just for the regions of interest, just for the cells that we could get a good signal from. Just in that data set that I was showing you that's kind of undersampled. So that's over 5,000. And that's just for one imaging beam and one plane. This is another slide to give you an idea of scale. So these are some other common neurobiology model systems. So C. elegans is a very simple worm that has about 300 neurons, and that can fit completely within our field of view. We could actually have a C. elegans. We've talked about doing this experiment a little bit, just having a C. elegans move around in a petri dish within the field of view and watching it. Yeah. I'd like to be able to, I mean, yeah, what I'd like to be able to do is maybe even 8 millimeters. No, the cortex only goes down about 1 millimeter, but then there's other brain structures that are subcortical. And we can talk about the prospects for that. For TwoPhoton, this is basically what you can do. There's nothing in this system that it's limiting our imaging depth. The limits to imaging depth, I mean, it's largely a function of staining intensity and sparseness. So if you've got bright signals that are really sparse, it makes it easier to image deep than if you've got densely labeled tissue that's dim. But we're basically running into fundamental limits of TwoPhoton, so photon budget, things like that. There are methods like 3-photon imaging that can help you get a bit more depth, but those are still pretty slow because the laser technology is not quite where it needs to be yet. And it also takes a lot of power. Yeah.
Have you established as to what adequate depth one should go into, at least insofar as that visual activity is concerned?
So you start seeing cell bodies for layer 2, 3 cells. So there's the cortex is a 6-layered structure. The first about 100 microns, 150 microns, in mice anyway, is relatively devoid of cell bodies, which is what we spend most of our time imaging. And then the layer 2, 3 cells start about 150 microns down. Once you get down to like 500 microns, you start running into layer 4, and then you get to layer 5, about 700 microns down. It depends on what layers you want to look at.
But do you see that the activity is sort of decreasing?
No. So, well, if anything, layer 5 is the most active. Layer 2, 3 is one of the more, has sparser activity than the other layers. Layer 4 is higher. Layer 5's the highest. Layer 6 is kind of low again. But it's not as if there's more coding going on in other layers. It's all acting in concert, and there's a lot of interplay with thalamus and other cortical areas. So we need to understand what all the layers are doing. Yeah. So starting, there's juicy things to study starting even 150 microns down. Then just to finish up this slide. Well, actually, to finish up your question. So 3-photon imaging is one thing you can do. Another thing is there's some really exciting work being done on imaging into scattering tissue. So aberrations you can kind of correct for with adaptive optics, kind of like what they do in astronomy. They try and correct for aberrations that you get from the atmosphere. You can correct for aberrations in the brain. And but for scattering, that's thought to be a bigger problem, or it certainly is a bigger problem because it's not as stationary as aberrations are. But there's actually been some really fast and exciting advances in imaging through and into scattering tissue. And I think we'll probably see some progress on that in the next few years. It's still, the decorrelation time is still within milliseconds. So it's a hard problem to solve. But from a physics standpoint, I'm not sure if it's an impossible problem to solve. We might actually be able to get some progress on that, and then that would let us image much, much deeper. So yeah, I talked about C. elegans. Zebrafish are sort of their juvenile state. It's fairly transparent. They're kind of small. We actually just got funded for a project to rapidly track zebrafish swimming around in the field of view and imaging neural activity. The Etruscan shrew is actually one of the smallest mammals. They eat insects, so they've got some interesting behaviors, and they've got this really small brain. And one entire hemisphere of their cortex would fit in the field of view. So that's another thing, another model system that we talked about. This field of view, like I said, this worked better than I expected it to work. This already is 50-fold, more than 50-fold greater in terms of area than commercial systems and the kind of 2-photon imaging that I was doing before. And since this 2-photon imaging is a point scanning technique, it also takes 50 times longer to image that, which is a huge problem. We can't sit there and image every single pixel location in the entire field of view and get the time resolution that we need. We need time resolution on the order of multiple frames per second, ideally tens of frames per second. And that's because that's the time scale over which patterns of neural activity are changing. The indicators that we have decay times that are around tens to hundreds of milliseconds. So even if we're not right there on them when the calcium increases in a neuron, it has an exponential decay tail that lasts tens to hundreds of milliseconds. So that's what we need. We knew that this would be a problem ahead of time, so we built into the system a way that we can image in 2 different regions at the same time. We do that using temporal multiplexing. We split the beam into 2 beamlets, and then we steer those beamlets to 2 different locations within the larger field of view, and those are simultaneously raster scanned. So here I'm showing imaging in primary visual cortex and imaging in area AM at the same time. Importantly, these are repositionable in X, Y, and Z randomly. We can even have them right on top of each other, image the exact same cells, image in 2 different layers, whatever configuration we want to do. And we can do this temporal multiplexing because the excitation laser is pulsed. It's pulsed at 80 megahertz. So what we do is we split it into 2 beamlets using polarization, and then we delay one by 6.25 nanoseconds. And then before we recombine them, they're deflected at different angles, and that angle of deflection is then focused on the raster scanning mirrors. So, and it's basically just showing this, so that we've got, well, actually, it's easier to see on the next slide where I'll show you the system layout. But first, real quickly, I'll talk about the demultiplexing scheme. So it's fairly easy to point these 2 beams at 2 different areas. For demultiplexing, then we need a synchronization signal, which we get from the laser, so we know when the laser is pulsing. And then this synchronization signal, when it's high, we count all the events to pathway 1. When it's low, we count all the events to pathway 2. So that's fairly straightforward to demultiplex. There's a number of different ways to do this, and I'm happy to go into detail. What we're doing right now is we actually take, so we've got our synchronization signal. We send through, we send it to one detector, and that goes into a veto signal in our counter board. Our signal from the PMT is amplified and then split into two inputs, and one is delayed by 6.25 nanoseconds. So instead of switching which channel we count from, we just delay the two channels relative to each other and veto pulses from them synchronously. And then we can either send this to digital counters or we can send this to low-pass filters and do analog sampling. I'm happy to talk about that in more detail if anybody's interested. But the catch with all this is it has to run at least 160 megahertz. So we do most of this with outboard electronics rather than trying to sample the signal and doing it rapidly in software. So this kind of gives you a layout of what's on the table. We have a titanium sapphire laser that's pulsing at 80 megahertz. Pulse width is about 150 femtoseconds. There's a photodiode from a pickoff mirror that we use for the synchronization signal for the demultiplexing. This is a half-wave plate, a polarizing beam splitting cube, a beam block just to attenuate total power to the system. There's another half-wave plate. These are both on motorized rotation mounts, a beam expander and a polarizing beam splitting cube. This half-wave plate rotation sets the ratio of power to the two beamlets. Then we've got the P polarization, the S polarization. The S polarization goes to a delay pathway that is 6.25 nanoseconds times the speed of light equals 1.87 meters. So that's the extra path length for this leg. We also have a tunable lens here, an electrically tunable lens that we can actually change the divergence and convergence of the laser, and that lets us independently control the Z depth that we're imaging, so the actual focal depth. And then to control X and Y, we have a steering mirror. So this is pivoting around a central point on the mirror. This goes through a relay system. Importantly, this relay system we learned needs to be asymmetric, because we have a beam recombination cube in here, another polarizing beam splitting cube. This is 2 inches on a side. The first way that we did this, it was symmetric, so it was actually coming to a focus at the interface between these two cubes. Turns out to be a really good way to cook your polarizing beam splitter. So we ruined a couple cubes that way, and then we redesigned this so that it's now asymmetric, and the focal plane is a little bit offset from this interface. But this just relays the image of this mirror to the scan mirrors. We've got X and Y scanners that are connected by a symmetric afocal relay. Then we've got a scan lens, tube lens. These are both multi-lens systems. Dichroic mirror, the objective, and then these are a 2-lens collection system to send light to the PMT. And here you can see that how these multi-lens systems are laid out and about how many elements are in each one. Okay, so here's some more data from this system. So this is now multiplexing, imaging in 2 different cortical areas at the same time. This is not with a dye. This is with a genetically encodable indicator called GCAMP6. So these mice are genetically engineered to express a protein that is fluorescent, and it fluoresces more in the presence of a high amount of calcium. So again, we get a nice field of view. These are repositionable anywhere in the field of view. Here's just an example of 2 depths at the same time. So that's the data over to the right. You can see there's signal to noise for the 2 different regions down here in the bottom. So this is showing you from the dorsal view, both regions are in V1, but they're separated in Z by about 50 microns. So they're imaging different cell populations within the same column. In the same experiment, we can reconfigure this as much as we want. So these are just a few different examples of moving these areas around. The steering mirrors that impose that first angle to direct the beamlets to different portions of the tissue are motorized, and so we can move them wherever we want. We originally had this kind of home-built servo-driven system, and now we've switched to these voice coil ones, so we can actually move really rapidly and reproducibly to different regions. Here's an example experiment where we're measuring in retinotopically matched areas in V1 and AM/PM, and we're showing a naturalistic video for this particular experiment, and these are just some example traces. And then in a different, for the same cell, same configuration, we showed a different visual stimulus, and these were black and white bars of different angles called gratings. And then we measured the activity correlations for, we had 150 neurons in V1 and 80 neurons in AM/PM, and we just computed the pairwise cross correlation. So we took their activity traces, we deconvolved them into an estimated spike rate, and then we computed the zero-lag cross correlation. And this is something that we had never been possible before with any other methods for the mouse, looking in two different cortical areas like this for such large populations. Even if we had put it in electrodes, we wouldn't have gotten this many units that were isolated.
So have you been able to distinguish or detect the differences from, at least in the orientation space and all that?
So the orientation space, like orientation tuning?
No, as you vary the orientation, for instance, or perhaps the location of things, have you been able to kind of detect that in the brain activity that you've detected or not?
Okay, so basically trying to do the decoding problem. Right. Yeah, we're just starting those experiments. So we're using a generalized linear model approach to fit the data and describe it. We've got coupling coefficients between neurons and different cortical areas. It's really early days on that. Frankly, we're working on fitting problems right now. So yeah, but that's exactly what we want to do. I had fun listening to your students give talks this morning on this. I think you guys gave me some ideas of some stuff we should try. This is just showing that we could actually see that the correlational structure changed depending on the stimulus. So this can give us hints as to what these different cortical areas are computing. But this is a really kind of first order or zeroth order experiment to do, and we're doing now the kinds of experiments that Hamid does, trying to decode the patterns of neural activity. Yeah. What was that? Oh, we haven't looked over time, I guess. They are reproducible. We get reproducible patterns of activity. So that was the first thing we had to make sure that we do, right? And in these experiments, we have to make sure that we're looking at visually evoked activity. So if we show the same movie 20 times, we should usually get similar patterns of activity, and we do. Does that answer your question? Yeah. Go ahead. So we're trying to do it over a short enough period where there's not too much, synaptic plasticity happening. However, we do expect, and we plan to explore, more kind of moment-to-moment changes in effective connectivity based on stimulus statistics and the behavioral state of the animal. So I showed you some behavior earlier in this talk where they were freely moving and using a touch screen. We're now training mice to do these head fixed underneath the microscope so that we can look at patterns of activity while they're getting visual stimuli and deciding which, they register their response by licking lick ports, deciding which lick port to lick. Well, for the time constants there's short-term plasticity that doesn't typically involve insertion of receptors or new proteins into synapses, and that can happen on the millisecond time scale. And that has to do more with release kinetics of synaptic vesicles and excitability. There's also for the types of synaptic plasticity that involve the insertion of receptors. Usually that takes more on the order of seconds to actually get the induction and to actually stabilize those new protein structures. It may take tens of minutes and for a change that can last hours to days. Okay, so I've talked a little bit about scanning. So speed, the major thing that we do is multiplexing, and that lets us sample activity from two different areas simultaneously. There's a couple of other things variations in the same thing that we can do. Resonance scanning, quad region scanning, arbitrary line scanning. I'll talk about each one of those. So resonance scanning is basically replacing one of the galvanometer-based scanners with a resonance scanner, which oscillates at a frequency, at a fixed frequency. You can change the amplitude, but you can't change the phase or the frequency. But if you can adapt your imaging to that particular resonance scanner, then you can get about a 4-fold increase in frame rate for a given number of scan lines. So this is data that was taken at 30 frames per second. That's resonance scanning. There's another variation you can do. You can do quad region scanning. So here we've actually got 2 different depths, and then we just interleave. So we're not doing this simultaneously. We're doing 2 different depths simultaneously, but then we just switch for interleaved frames between these 2 different regions. And this gives us 2 different depths in 2 different areas. So that's a way that you could study processing at 2 different cortical layers and 2 different cortical areas at the same time. Here, we've taken 2 different areas, and now we're just switching between those 2 different configurations to sample from 4 different areas. They're at different depths as well, but they're not lined up on top of each other. So that's 4 different cortical areas. We can also do arbitrary line scanning, where we just, within this larger field of view, we just pick a scan path instead of raster scanning, which is really inefficient because you end up spending a bunch of time sampling activity of blood vessels, which have no activity. You can actually have a directed line to scan. You can get nice signal to noise with this. One problem with this is that if the mouse moves, you can't post hoc do any registration. If your raster scanning is fast enough, you can often offline, after you collect a movie, do image registration. A lot of times just rigid body image registration can buy you a lot in compensating for movements when the mouse is licking or registering the response or otherwise moving. It turns out that the signal to noise for these indicators is so big that it's actually not too badly affected by movement. So here's just an example where we've coded movement manually by imaging the mouse. Black is stationary. Yellow is kind of a grooming motion where they'll bring one paw up and kind of rub their whiskers. And a red is a very forceful movement where they're actually trying to maybe torque themselves against the restraints that they're in. And you can see that those cause changes in fluorescence, but they're really small compared to the fluorescence transients. These are shown at the same scale, 200% delta F over F. They're relatively small, and they tend to be negative, not positive, because usually a structure in a region of interest is moving out of that region rather than moving into it. There's the background's black; it's not white. So if there's any kind of misalignment, it tends to be negative rather than positive. So it's actually plausible that you could use this approach in a behaving mouse as well. So I've shown you that there's a lot of flexibility in this approach. This is, like I keep saying, this worked better than I expected it to, and we've been lucky to receive some additional grant funding so that we can kind of take this to further generations. This is a system that we're building right now, and we've relaxed the constraint of using off-the-shelf lenses and have moved to completely custom lens systems. This is a system that is going to give us a greater than, this is actually just the scan engine. This isn't even showing the objective. This is going to give us a greater than 5 millimeter field of view and a greater than 0.5 NA. And we've also decoupled the scanning. So we no longer simultaneously scan both beamlets at the same time. We can now actually independently scan them. So one beam can actually scan a full field of view, and then the other one can scan one small postage stamp if you want. So we've got 2 optical relays here for X mirror, Y mirror. This is a scan lens. X mirror, Y mirror, scan lens, and then the beam recombination happens between the scan lenses and the tube lens, and it's based on polarization as well, so you can actually have the exact same wavelength of laser coming in both, and then this is a multi-lens tube lens system. So this lets us scan. This is an improvement in a couple of different ways. It gives us more flexibility, and it also lets us scan a larger beam over a larger angle. And these are huge optics. This is just to give you, we've received some of it in the mail. This is a $20 bill just to give you an idea of scale. This is just the tube lens assembly. Yeah. So this work was done, most of this was done by Jeff Sturman here in the middle. And we couldn't have done it without Mike, your guys' colleague, Mike Kudinoff, who really taught us a lot about optical design, tricks of the trade, and strategies to rapidly generate this imaging system and get the results that let us know that this could be done. So I want to acknowledge Jeff and Mike again and our funding sources, and I'm happy to answer any questions. Thank you very much.
We have time for some questions. Well, you've been asking questions the whole time, so why not? Or have you run out of? How big of, was the subject that you can work on as far as your machine is concerned? How big of a subject can you handle?
Well, there's no reason you couldn't use this on, yeah, you could use it on a human, but you'd have to stain the tissue, right? So you'd have to use you'd have to either genetically engineer the human, which we don't really do, yeah, or you could use a dye, a calcium dye, and inject that. More proximally and nearer term, using something like this in a non-human primate. So they're getting better at using viruses to express transgenes in in a non-human primate brain. And I've talked to colleagues, and we're working on grants and stuff to adapt some of this for that. So one thing I didn't talk about is we have a design for a water immersion objective, and we're going to send that out for quoting here soon. But most of what we've been doing to date has been air immersion, which it has some drawbacks, but it has some benefits for ergonomics of these experiments, and that you don't have to maintain a gigantic water well since these objectives are so large. And one of the things with non-human primates is that, for mice, it's nice. The mouse can sit there, and their visual cortex is kind of on the top of their head, so they're pretty comfortable just sitting there while we image straight down on top of their head. But for primates, the visual cortex is back here in the back, and they have to be kind of sitting in a chair, and getting the objective around at that angle is doable. It's an optomechanics challenge, but it's doable. But what you really don't want to have to do is try and find some way to maintain a water bath while you've got an objective coming in and up at an angle at a primate's head. So the air immersion buys a lot for that. You can also tweak the working distance. These are really long focal length systems, so that gives you a lot more working. Makes it easier to have a larger working distance, which also makes it easier to deal with some of these non-human primate preps. So that's probably what's going to happen first.
Have you been able to, for instance, to measure, do an A/B comparison between what, the visual field, the mouse or whatever you're testing is versus an obstructed site? In other words, they would have their eyes, their visual system completely obstructed, and see what the difference would be.
Well, I mean, we've done an experiment like that with intrinsic imaging years ago, but I mean, the take-home is that you just have spontaneous activity, right? There's no patterned activity anymore. You can get, if you've all had the experience, you can close your eyes and kind of look around the room, and you can kind of tell where the lights are, right? You get kind of a light-dark signal. So if you have some sort of periodic, like a drifting grading, black and white bars, sometimes you can get some really kind of diffuse activity that's periodic. But yeah, you don't have much in terms of patterned vision.
Any other questions? Yes.
Have you guys talked to anyone that started approaching like in vitro systems or non-brain type imaging systems where you want a large field of view or something else?
So that's a great question. So I kind of told you the story why I came to this, and actually, fortunately we got these grants, but some of the critiques that we get on our grants is like, this is a really esoteric thing to build. But we also got the opposite comment on some of our I actually had one grant where the comment was, this is impossible. If it was possible, then Zeiss or Olympus would have already built it because the demand certainly exists. So it's kind of, people kind of fall into two camps about whether or not they're excited about this. I don't spend a lot of time thinking about other particular applications for this, but I always, because I'm not really quite sure what other problems would fit in this regime where you need this time resolution over a spatial scale that's millimeters and a spatial resolution that's, a few microns, or 1 micron in X and Y, but 12 microns in Z, which we can make a little bit better. Yeah. So one thing that's come up is there's a guy who wants to look at this for cell incorporation into larynx, and wants to look at that over a larger spatial scale and wants to look at dynamics of those cells. I've been told that there are interesting problems in the Arabidopsis root system and calcium signaling in the root system. Again, this is a system that...
Welcome to Unified Conferencing.
Don't worry about it. Thanks.
You are the first person to join the conference.
So it's a... the root system spans millimeters. The dynamics are in the millisecond time scale, and so they want systems like this.
But particularly, I was interested in looking at the other side of your image, which is the blood vessels.
Okay.
So if we're saying things in there, looking at those networks, flow and pulsatile flow.
Oh yeah, no, that's a great, yeah. So there are labs that do that. So there's actually a lab in UCSD that does a lot of this, and they use two-photon microscopy as well. They're still using stock objectives because they can get the resolution that they need. They don't need 0.4, 0.5 NA. They can do a lot with 0.28 NA, and so they're using some of these just kind of commercial objectives. But they've solved some interesting. They had a paper not long ago with an interesting scan engine that they're using. But yeah, it's I guess for maybe more like capillary bed, really small blood vessels, you could use this. Yeah, absolutely. Yeah.
Have you released any data sets or are you thinking about it?
Oh, absolutely, we'll release data sets. So one of our, this one, the grant that we have from Simons, that's a big component of it. So that particular grant is to understand neuronal dynamics across multiple brain areas at the same time, and contingent on funding is the idea that you're going to release data sets to people. So we're working on it. We've got some of the, we want to make sure that we're releasing high-quality data sets. And actually just day before yesterday, I saw some of the most reproducible stuff that was coming off of that system. And so we'll be trying to do that as soon as we can. Absolutely. And all the optical designs, that's all open source. So actually, we did a bioarchive preprint in 2014. Has my name, Mike's, and Jeff's on it. It's not too hard to find. That's been downloaded over 4,000 times. That has all the optical prescriptions. And then it was just recently accepted to Nature Biotech, and so hopefully that'll come out in print before too long.
What did it cost you to build it?
So the system that... No, it's an important consideration. And one of the things that we're excited about for that particular manuscript that we did a lot of stuff with off-the-shelf lenses is it makes it much more doable, right? So the custom lenses in it were a few thousand a piece. And I mean, the biggest expense is the laser itself, which are 100, 150K for a titanium sapphire laser. But yeah, you can build a system for this like this for in the neighborhood of 100,000. Less than well, I mean, just because you're building so much of it yourself. I mean, as far as if a company built this and wanted to charge you for it, it'd probably be about the same. It's not that bad. You don't have to buy a commercial objective, right? You don't have to buy a Trinoc. You don't have to buy any of those things. You're buying off-the-shelf lenses. The alignment process is not what I would recommend for everybody. I mean, we're a neurobiology lab, right? And we're trying to do metrology and stuff. And yeah, we're again, Mike was a huge help in making this doable. But typical neurobiology labs would not necessarily want to spend their time doing that. And so we're open sourcing all this stuff, but also anybody who wants to kind of make these assemblies, that makes it a lot simpler. But then doing these assemblies with some of these last systems that I was showing, this is expensive. This is probably going to be around $100,000 just for the optics. Yeah.
All right. Let's thank our speaker again.